for our speaker this morning, we're very lucky to have Dr. Benjamin Chen. Dr. Chen received his MD from Cornell and completed his PhD studying the HIV virus with Nobel laureate Dr. David Baltimore. He has now been studying HIV for nearly 30 years, the last 17 of which he's been at Mount Sinai, where he currently serves as the Irene and Dr. Arthur M. Fishberg Professor of Medicine, as the Vice Chair for Research at the Department of Medicine, and as an Associate Director for the Medical Scientist Training Program at the Icahn School of Medicine. He maintains an active research lab, is a member of the American Society of Clinical Investigators, and is a consulting editor for the Journal of Clinical Investigation. Please join me this morning in welcoming Dr. Benjamin Chen.
Thank you, Dr. Wilmer. And thanks, everybody. Thanks, Dr. Thomas, Dr. Murphy. It's really nice to be able to have the opportunity to give grand rounds. So what I thought I would do today is spend some time sharing with you the research that my group has been focused on in the other epidemic, and that's the epidemic and pandemic of HIV. And a lot of the work that we have focused on in the last 17 years, I guess perhaps more last 10 years, we've been really using methods to visualize infection with microscopy and other tools to try and understand the complexities of the pathogenesis of this virus, which is very difficult to cure and to eradicate. So I'm going to tell two stories, and the first is a long story where we've used imaging to look at the cell-cell interactions that help facilitate HIV infection and make it particularly difficult, that facilitate immune evasion and make it particularly difficult for the immune system to clear, we think. And then the second part of the talk will be an entirely new project in the lab, which is focused on understanding and visualizing, identifying latent cells, the latent cell reservoir, which has really been a challenge in the field. And hopefully it's all new data. And I'd like to be able to share with you an approach which I hope will lend a new perspective on how HIV lays dormant and may have some implications for cure as well. And so shown here on this introductory slide is a live image of a cell-cell interaction, which we think is likely very important for spreading HIV between immune cells, called the virological synapse. Okay, so I have no disclosures to report. Okay, so we are still in the midst of an HIV epidemic. I think it's helpful to review the global statistics, particularly when we're in the midst of this coronavirus pandemic. There are 38 million people estimated to still be infected worldwide. That's 1.7 new infections per year, which is still quite an alarmingly large number. And 0.7 million deaths per year attributed to HIV. It still impacts the particular areas particularly dramatically, so South Africa, but here at home, there's over 1 million people who are still living with HIV. And because there is no cure, and this is a chronic infection, for which we do now have treatments, but there is no cure, that the incidence of HIV globally continues to increase. So you see here, this is a trajectory which we have not been able to bend the curve for HIV as yet. But this is an entirely different timescale from the pandemic that we're fighting right now. So the natural history of HIV infection, which I think everyone is familiar with, involves an acute infection, which is a flu-like illness. And HIV infects as the CD4 T cell, which is the helper T cell, a central coordinating immune cell, and it infects it and winds up depleting the CD4 T cells. And during acute infection, there is frequently a drop in CD4, and if untreated, the immune response, there is a rather robust immune response to HIV, which does in part control control the initial acute viremia, but it doesn't eliminate viremia. And it's this persistent viremia over the course of years in the face of which is able to evade a rather robust and immune system, which leads to the deterioration of CD4 cells, and over time results in the opportunistic infections and death if untreated. And this is driven by a virus that has, as a part of its life cycle, so this is the virus particle here. It's an RNA virus. It binds to the cellular receptor CD4, which through its viral glycoprotein envelope, and it fuses with the plasma membrane. And it's the RNA, whereupon the RNA is reverse transcribed into DNA, and it inserts into the genome of the target T cell. And it's this covalent insertion of the viral genome into the host chromatin, which is irreversible, which can lead to a latent state, which makes the virus particularly difficult to eradicate. So unless that cell dies, a copy of the virus lives on in that T cell for the life of that cell. And then what happens in this process is that the viral gene expression program proceeds, and virus is produced from the plasma membrane. This free virus can come back, and as we've seen in textbooks, the model often is that the cell-free virus comes back and attaches to another cell and restarts the process. And fortunately, we have at our disposal now a host of drugs which target key enzymatic steps or key binding steps in the virus life cycle. And these together over the years have resulted in the ability to therapeutically treat HIV. And so shown here is an early study where treatment of HIV resulted in the dramatic decrease in viral load and subsequent restoration of CD4 counts. However, in the absence, so long as we're taking highly active, the patient is taking highly active antiretroviral therapy, viral loads can be suppressed. But however, if treatment is interrupted at any point, this almost inevitably leads to viral rebound. And so it's thought that really the virus cannot be spontaneously cleared from infection. And so we've been interested in particular in the reasons why it's so challenging for the immune system to eradicate or to control HIV, except, and in part, we start from first principles of virology and tried to understand not only the mechanisms of how viruses infect individual cells, but try to put this in the context of the immune cell interactions. And so here we've known, for instance, for many years that dendritic cells can bind to virus without themselves getting productively infected. And this can lead to even a much more enhanced infection when the dendritic cell interacts with a T cell. And more recent, subsequently, in cell-cell interaction between infected CD4 cells and uninfected CD4 cells can also lead to more productive or more efficient infections. And so this has been the focus of my lab for a number of, for many years. And in 2004, there was a description of what was called a virological synapse, which involved the binding of an infected cell with an uninfected cell. And at these structures, what the authors noted was there was an actin-dependent relocalization of the viral receptors, so the envelope protein, CD4, the target cell receptor, and the Gag protein, the viral structural protein, to the site of cell-cell contact. And so this implied that there was some active processes which T cells, in which T cells participated in the infection of one another. And so this, and what we set out to do at the time was to develop a way to visualize this infection process. And so we developed a virus which carries the green fluorescent protein cloned into the core structural protein Gag, and we called this virus HIV Gag-IGFP. And the green fluorescent protein, when it's part of the structural protein, can create intensely fluorescent, green fluorescent HIV particles when produced in cell culture. And so what we could do, what we did in this initial experiment was to take this fluorescent virus and sprinkle it on CD4 T cells and compare the efficiency of that process with a CD4 T cell, which was actively expressing that same green fluorescent virus. And so we were comparing the efficiency of infection when the cells had the opportunity to interact versus the cell-free virus that is independently put on the cells. And shown here is a FACS plot showing the fluorescence of the recipient cells when you dump on a lot of virus, just a short co-culture in this case. So this is not active replication, but it's just how much virus is being absorbed to these target cells. And we found that even if we put on a large excess of virus, very little fluorescence shift was occurring. However, when we co-cultured these infected cells with uninfected cells, we saw a very large fluorescence shift on this this dot plot. So indicating that these recipient cells, which were dyed red to discriminate them from the infected donor cells, they had taken up an enormous amount of that fluorescent virus. And then we showed that that fluorescent shift was dependent on an intact envelope protein. So it was driven by the interaction between cells was driven by the viral envelope glycoprotein. And so the envelope protein seemed like it's mediating, has a function almost as a cell-cell interaction receptor in this case. And the interaction was leading to more efficient infection. And so the nice thing about green fluorescent protein is that it provides a nice way to visualize this process. And we can visualize it not only in fixed cells here. So this was an infected cell expressing this green fluorescent virus, and 3 target cells, which are dye labeled red. And we saw in these early images accumulations of green fluorescent protein concentrated at the sites of cell-cell contact. We could do time-lapse imaging. And so shown here is another infected cell. These are all primary CD4 T cells. And you can see that this cell from up here just crawled in and interacted and now is stably associated with this donor cell. And and this interaction is can be monitored over time. You notice that there's not much green fluorescence at the site of cell-cell contact. And so if we measured and we followed a lot of these cell-cell interactions over time, what we noticed was that initially the viral structural proteins were not at this site of cell-cell contact. But over time, they would be, in many cases, recruited or accumulate over time at the site of cell-cell contact. And so we could count this and find that in most cases, over time, we saw a adhesion first, and then we saw accumulation of the virus at the cell-cell junction. And if we used some faster video microscopy approaches, this is a method called spinning disk microscopy. And so you can achieve frame rates of about a second per image. And this is a 3D image. You could see here a synapse, which is already formed. There's a target cell over here. And we were really astounded to see how much viral material was being internalized or transferred into the target cell. And so, and there was an active process of recruitment and depletion of the viral structural protein from an area surrounding this cell-cell interaction zone. This is all actin dependent, so it relies on cellular motors. And this is a process which, and you can see that some vesicles being recruited into this. And so this suggests that the virus is really utilizing a lot of cell biology, a lot of the active processes to assemble viruses at this site of cell-cell contact. And then there's an active process of internalization on the recipient cell. If you look when we look with electron microscopy, what we saw at these cell-cell junctions were these viral presents assembled at the site of cell-cell contact. These are budding forms. These are budding particles that are about to be released from the cell. So even before that virus particle has been entirely formed and released from the cell, it's already interacting with a recipient cell. And just looking at these images, you can also appreciate that this process is occurring at the cell surface. It's likely that antibodies may still be able to interact with that, but it is a pretty tight association. So we were interested in understanding how antibodies would interact with these structures, which we'll get into in a little bit. With longer duration imaging, this is a image analysis of over 48 hours, over a couple days. You can see this cell here, which is infected, interacting with its uninfected cell. And this is all in cell culture. And about 32 hours later, that recipient cell becomes infected itself. So we can see that, and in this particular experiment, we could trace all the newly infected cells to an interaction event with an infected cell. So at least in cell culture, it was critical in this model for the cells to interact, for them to become infected. That was all in vitro. This is actually, we've also developed methods to study this in vivo in humanized mice. And so if you take an immunodeficient mouse, that's that can be a receptor for a recipient for xenografts of human either peripheral blood mononuclear cells or of hematopoietic stem cells. You can grow a human immune system in that mouse and conduct intravital imaging on that in that mouse. We can take those similar green fluorescent virus and infect that mouse. And here is a virus which is expressing GFP not in the context of the structural protein, but just as a free protein. So it fills the cell. And what we were surprised to see is many different morphologies. Some infected cells are moving very rapidly. Some are really restricted in their motility. And they had these elongated processes, some of them, and that might be indicative of cell-cell fusion events or syncytia. So so T cells are, I mean, I think we can all appreciate that the function of our immune system, and particularly the cellular immune system, really is to conduct immune surveillance. These cells are constantly wandering in search of their cognate antigen, and this is an image of a window on the spleen. And so you can appreciate that these infected cells are potentially interacting with many other cells. So what we set out to do is also label some of the uninfected cells and see how they interact with the infected cells. So in this case here, we have two different color viruses. We have a green virus and a red virus. We noticed that the green viruses tended to sometimes cluster around certain areas, and the uninfected cells could wander through these areas. The motility of these infected cells is much more restricted than these uninfected blue cells. So uninfected cells are unrestricted in their movement, whereas the infected cells often have discoordinated movements and are more confined to a very small area. And so we also set out to image this process with a greater number of uninfected cells, so we could see if we could capture uninfected cells interacting with infected cells within a humanized mouse spleen. And so if we genetically label some peripheral blood mononuclear cells, and these are CD4 cells from a donor, and we inject them into the mouse at a higher frequency, these are uninfected cells within a humanized mouse spleen. And then if we look at a similar mouse which had an infection with a red fluorescent virus, we can see that well, we appreciated that there was more uninfected cells clustering on top of these infected cells. If we zoom in on some of these, we could see that there were frequently cells that were paused at one end or the other of the cell. And so we could measure this, the mean velocity of cells. So we just track them and see how fast they crawl. And if we chose a region of interest away from infected cells, the mean velocity is faster than when cells are crawling on top of these infected cells. So the infected cells are having an impact on the uninfected cells that are conducting their normal immune surveillance. And we think that this, although it's, these are not as long-lived interactions as we've observed in cell culture, we think that these may be sufficient to promote the spread of virus in an in vivo context, in an animal model. And so, so this is our model, which is for cell-cell infection. So there's a CD4 envelope adhesion-driven adhesion. There's recruitment of envelope, which I won't have time to go into, but we have methods for fluorescently labeling envelope as well. And we think this is a two-step process where envelope is recruited first, and then Gag, the structural protein, is recruited. There's transfer of this material into the recipient, and then productive infection. And really, what this process really involves is active cellular machinery on both ends. This is an actin-dependent process. There's an endocytic process, which appears to occur on the recipient cell side. I think these are and this involves like a highly concentrated a level of virus that's transferred directly to the target cell. And so these are aspects of the virus interaction with T cells, which are not necessarily fully integrated into our understanding of pathogenesis. And so one of the key questions we had was to what extent are these synapses vulnerable to neutralizing antibodies? Is this one of the ways, another way that the virus uses to escape host immune responses? And so over the years, we've conducted a few studies to measure the impact of antibodies on cell-to-cell infection, and compared that with the exact same virus in a cell-free form. And so we have assays where we can use the same virus. It has the same glycoprotein on the surface. So presumably, you know, the gene, the protein is the same protein produced by the same virus. But we can look at it, how it interacts with or how it's neutralized by antibodies when it's presented in a cell-free form, when the particles are just sprinkled on the target cell, or if it's presented as an infected cell infecting an uninfected target cell. And so I'll show here is a bunch of different plots. So some antibodies, this is an antibody which binds to CD4, the target cell. And so the CD4 receptor is on, because it's on the target cell, and we're blocking the ability of envelope either from the virus or the cell to interact it, it blocks both forms of infection equally. However, if we use other antibodies, this is these three are anti-envelope antibodies. These are monoclonals, which are very well characterized with regards to where they inhibit 17B binds to a CD4 induced conformation on the envelope glycoprotein. There's about 100 or almost a 2 log difference. It's less efficient at blocking cell-cell infection with 17B. B12 also binds to the CD4 binding site. Here's another shift of about 10-fold, and 4E10 binds to GP41, the transmembrane subunit of envelope, and there's a relatively large shift there. We went on to look at how cell-cell infection could be inhibited if we used transmitted founder clones. So those first studies I showed were with laboratory adapted strains, which are well known to be easier to neutralize. And so here we developed, Hung Ru Lee in the lab developed a series of transmitted founder clone envelopes and clone them into this fluorescent virus system so we could measure cell-cell infection. And first, she tested these primary isolates. These are from acutely infected individuals. These are known to be more difficult to neutralize in general. And first, this test is with HIV immune globulin. This is antibodies purified from HIV infected patients and pooled. And one can see immediately that there's many, like the blue lines are cell-free virus, or the red lines are cell-free virus. And the blue is cell-to-cell infection. Very little cell-to-cell infection is neutralized by this HIV-IG. Now this is in contrast to that there is some activity against cell-free virus. Now this also demonstrates or illustrates a problem with HIV immunity, in that even though these patients have, this is relatively high titer immune globulin, it does not neutralize cell-free viruses from primary isolates particularly efficiently. However, if you'll note, we noted that the cell-free virus can be neutralized. There is, at higher concentrations, you can detect neutralizing activity against many of these strains. And so we went on to look at a whole host of different, more potent monoclonal antibodies. And these monoclonal antibodies, in some cases, could exhibit, like in this case, a 3,000-fold shift in inhibitory potency. So this is a incredibly large, dramatic difference between cell-to-cell infection over here, where it requires, the IC50 is in the tens of microgram per mil here, versus hundreds of nanograms. This is a very large shift. We also noted that in many cases that at the highest titer, these viruses, these antibodies rather, could not neutralize the cell-associated inoculum to 100%. So it had decreased potency as well as decreased efficacy. It couldn't reach 100% efficacy, even at the highest doses in many cases. And this pattern, there was a different pattern for each clone. So on the top, we have two primary isolates, the columns here. And then here on the right are two lab-adapted isolates. The lab-adapted isolates are a little easier to neutralize. They don't always show the same shifts in differences in potency. And so this is very strain-specific and epitope-specific. So this is a monoclonal against a V3 glycan. And here's two of them with V3 glycan reactivity. These are rather potent, newly generated, broadly neutralizing monoclonal antibodies. And there's a big difference between how well they neutralize cell-free versus cell-to-cell infection. And so shown here is just a little heat map summary of a bunch of monoclonals against different targets on the left, those four different isolates across the top. And, you know, in many cases, this red indicates, you know, often thousand-fold or hundreds of fold differences in inhibitory potency. This is an IC50 calculation. And so what we think this is suggestive of is that that primary isolates, well, we know that primary isolates are more difficult to neutralize, but this is particularly so for cell-to-cell infection. And potent, we noticed both changes in the potency, so they had a lower IC50, they were less efficacious as well against cell-associated HIV. And this dependence, there was high dependence on the antibody and the strain. And we think this is indicative of this is the same molecule in two different contexts. So we think there may be conformational differences in the envelope protein when it's on the context of a cell versus the context of a virus. And so we're working on further mapping these differences and seeing if we can test for vaccines which can more effectively block both cell-to-cell infection and cell-free infection. Okay. So that's the first story, and what I also wanted to do is take a moment to share with you some of our latest work, which is also, I think, described appropriately as an approach to visualize HIV infection, but in this case, to visualize it not only when the cell is actively infected, but also to identify latent cells which have turned off their HIV genes and are persisting as part of the latent reservoir. And this has been an important goal of HIV researchers for quite some time, but it's always been a challenge to identify latently infected cells, in part because whenever you, to do so, to define it as being latently infected, very often this involves activation of the cells, right? So you need to activate it, see if the virus is intact, it's in the cell. Otherwise, and that process actually perturbs the latent state. So if you want to study what allows a virus to maintain itself as a latent virus, you need to figure out a way to do it without activating the cell and perturbing that system. And so this is the idea here is to study cells in this state, which are known to be transcriptionally silent, or at least you know, if you take peripheral blood from a person who's efficiently treated on highly active antiretroviral therapy, and measure HIV RNA in those cells, there really won't be, you won't detect much HIV RNA. And yet, if you allow those cells to grow out, and if there's an interruption in therapy, there are cells in there which will lead to recrudescence of the viral infection. And the goal of cure research these days is to try to understand what the state of this infected cell is, which is latently harboring the virus, and to try and reverse that latent state. And so in order to understand what strategies will be effective, I think it would be useful to know to be able to identify these cells in an unbiased manner, and to test them with different agents. And ideally, if you had you could do a genomics profiling or transcriptomics of individual cells. This would be ideal. And the other approach to cure is, in addition to reversing latency, is to use cytotoxic agents, I guess, or immune, either antibodies or T cell therapy or even vaccines, things to heighten the immune response against these newly activated cells. And if one can do this under the umbrella of ARCH, continued antiretroviral therapy, ideally we could begin to clear this reservoir of infected cells. And there have been models which have been proposed that the reservoir is composed of cells with different decay rates. That is, when you have when you treat someone with antiretroviral therapy, there's a pool of cells which dies, which is eliminated by the virus replication. And since the replication has been eliminated, this pool of infected cells is immediately cleared. And there's different phases of decay of viremia. So this is viremia over time. And that there are different cell types which are still somewhat poorly understood, which are probably memory cells. They may be enriched in certain markers, but these are cells which are otherwise indistinguishable from other uninfected cells. And there are, at this bottom of this figure here, there are cells which have a decay of many years, and they are likely to be replenished by their homeostatic or T cell activation processes, which do not clear the virus. And so, so it'd be nice if we could identify these cells at the different phases during antiretroviral treatment. And so, so this is our approach. I'm going to show you some evidence which we think shows that we can do it. We can mark latent cells in a humanized mouse model, and then show you some what we've done to try to understand or to look at the profile of those cells with single cell transcriptomics. Okay, so this is a system. It's a two-part system which utilizes a red fluorescent marker that is sensitive to the Cre-lox reporter, the Cre enzyme. And so there's a promoter driving DS-red. It has these sites which can be recombined out. And constitutively, if there's no Cre in the system, this cell is red, and we mark CD4 cells with this so-called HIV-induced lineage tracing system. And then we do this with a lentiviral vector. And if we introduce the Cre enzyme, this will be recombined and switch to a green fluorescent phenotype. This is the little vector that we use. And we've created a virus which doesn't turn the cell green, and we've taken out the green fluorescent protein, and instead we've put in this Cre recombinase. So this is the key to the switch. So we put in the switch into the T cells, and then the virus that is incoming will induce recombination in that target T cell. And this virus is actually reasonably replication competent. If we have, so here's some cells. This is a FACS plot showing that we can introduce the switch into cells. This is DS red, and then when we introduce the virus, they switch to green. And so this is a FACS plot illustrating that the switch can occur. And this is an irreversible genetic switch. So even if the virus turns off and becomes latent, is transcriptionally silent, these cells will remain green. And so we developed this system in vitro, and then we also used hematopoietic stem cells. These are cord blood derived cells. We can transduce those stem cells with this red-green switch. We introduce it into neonatal mice, and then we have a bunch of mice which have a human immune system. They have T cells, CD4 cells, CD8 cells, and a good fraction of them express this genetic switch within their CD4 compartment. We can infect these mice. They have a viremia, and we can treat them with antiretroviral treatment in their chow. And if we can suppress their viremia with that. And if we look during acute infection of one of these mice, this is the spleen, the lung, and the bone marrow are showing that we're seeing green cells, which means that these are infected cells in this mouse, and we can sort them. And then we can take those sorted cells and do single cell sequencing. So this is a dimensionality reduction, so-called Tisney plot, where each dot represents a sorted T cell from this infected mouse. And these T cells are actually remarkably similar to T cell lineages. So they're in this, within this clustergram, within this Tisney plot, we can see an activated population of cells, T regulatory cells, T follicular-like cells. You can see some quiescent cells. So there are diverse lineages of T cells, and so what we did for this initial experiment was to look at where the infected cells are. And so these red dots represent HIV-infected cells. These are productively infected, so this is not quite latency yet. But we can identify what's unique about an infected T cell. We find that they are found in many different T cell clusters. So HIV can infect a diverse population of cells, even though it's known that in vitro you do need to activate cells. Those T cells may go on to become resting and quiescent, and may populate these different T cell transcriptional states. And shown here on the right is a figure or control looking at the expression of that red-green marker. And it's a little more evenly represented across the different populations. These two populations have more RNA in them, so we do see a higher fraction of these as being positive. But we're very interested to see how this distribution of HIV acutely infected cells looks like. It might be it's a different subset from all T cells, so it is enriched in certain T cell states. But this gives us an idea as to what how HIV may change the transcriptional state, or how it selects for infection of T cells with particular transcriptional states. So here's two mice, data from two mice that were treated with antiretroviral therapy. These are both suppressed below what was the limit of detection of our assay. We sorted green cells from the spleen of two mice, one treated for 10 days, another treated for 29 days. And we conducted a similar T cell profiling exercise. And the cells also clustered very nicely in this case into 7 different groups. We again found that although there are some differences, we saw a smaller number of what are now survivor cells. These are cells which have survived and have HIV in them by virtue, and we know that these green populations are enriched for DNA. And so we're assuming that this is indicative of cells which are have survived infection and persist after 10 days of ART or 29 days of ART. And we noticed that, for instance, there are fewer activated cells slightly at the day 29 in the persisting infected cells. There are some shifts. In this case, at day 29, there's also an increase in Tregs. The numbers, I think it may be that we're seeing some changes in where HIV is located over this early treatment phase. But what we can also do is, what's surprising here is that there are infected cells, again, represented in many different lineages. And so what we can do with these data, and we're beginning to do this is still a work in progress, is we can compare the infected cells with their uninfected bystanders and see what gene pathways are perturbed. Or we could look at the ART-treated infected cells and see what gene pathways are perturbed. And you'll notice here, I think the one that sort of sticks out is, although there's many which are common between the acutely infected and the ART-treated cells, like here on the top is this EIF2 alpha signaling pathway. This is a translation regulation signaling pathway. And we noted that this is upregulated in acute infection and likely downregulated in persistent ART-treated HIV-infected cells. So it's interesting that there are some pathways which are modulated in opposite directions when you're comparing an acutely infected population versus a latently infected population. So here is just a summary of a related comparison between day 15 acute cells and day 10 ART-treated cells. And again, this is there are some pathways which are regulated in opposite directions. And then there's some common pathways which appear to be common to both treated and untreated HIV-infected cells. And with this pathway analysis, so what does this all mean? With this pathway analysis, we can look at the driver transcription factors which are operative in these cells. And so there are certain key transcription factors, MYC-N being one, which, and MYC itself as well, which are regulated in opposite directions. There's some players, the T cell receptor as an upstream regulator seems pretty intuitive. And then there are some pathways which look similar to these genomic changes, which within a pathway analysis might allow us to begin to choose drugs which would modulate latency in a particularly effective manner. Sirolimus here is one which is already well known in the latency literature and has been tested as an agent which does not suppress HIV or transcription in the context of when you're treating actively infecting cells. And so there are some very interesting potential therapeutics that could be utilized for treatment. So how do we think about the latent reservoir given this type of perspective. Obviously, this is something we're continuing to pursue. There's a lot of work these days. This is a nice review from Yachi Ho, suggesting that the reservoir can proliferate and survive. And there's also homeostatic proliferation, and there's some effect of HIV infection as promoting proliferation in some of these cells. So what we want to do is take these types of this type of a picture and understand what states of transcription and phenotypic states, these latent cells, how these relate to these different functional states. And I think, so we have a system which can track latency. We have an ability, we think, to get a very fine transcriptional map, and we find that many different clusters of cells can support HIV infection. And this is true of both active infection, acute infection, and ART-treated cells as well. And what's ongoing now is longer treatment to see if latency is enriched in particular populations over time. And so what we would like to do is to better define these transcriptional pathways and hopefully identify drugs which can counteract or disfavor latency. And with that, I think I thank you all for staying tuned. There's a lot of people over a number of years who've contributed to this work. Namita Satija, Don Nguyen, and Forum Patel have performed most of this this latency marking system. Many of the others here listed here, some old lab members contributed to the imaging, and we have a very nice collaboration with some of the genomics folks on the single cell sequencing and latency marking studies. And I think I'll see if anyone has any questions. Thank you all for your for hanging in there, and hope you learned something.
Thank you, Dr. Chen. So we have time for a question or two, and I invite you to put your questions in the chat, please. Ben, you see that and comment in the chat?
Let me pull up my chat.
Oh, sorry. What Sachi do is mentioning she's getting lots of long hauler vibes from HIV COVID. So
I can unmute myself and speak.
Oh, yeah.
Ben,
Okay.
thank you so much. I just felt like, a lot of this like dormancy that we're seeing, the intracellular survival, the up and the down, I wonder if like that's where we're headed with COVID, because in our post-COVID care, we're seeing so many long hauler symptoms, and we don't know whether it's the virus that's still around or is it the immune system doing the up and down thing. So I found this talk extremely interesting in its own right, but I couldn't help because my brain is so occupied, as I'm sure everybody else is.
As we all are, yes.
Yeah.
Yeah, in terms of, I guess, inflammatory responses and COVID, I think there are a lot of parallels. Obviously HIV is, it's an inflammatory, the chronic infection is associated with chronic inflammation. I think with COVID, it's an acute infection, which is cleared relatively rapidly, but it's not so well understood why we have hyperinflammation and why it may persist even when local replication in the respiratory tract is undetectable. But I think what's interesting is that there is this fundamental immune activation, and I guess if you have chronic inflammatory issues in various stages of COVID disease, you know, I think it may be it could be indicative of you know, viral niches. You know, I think one thing is that COVID is much more compartmentalized. So HIV infecting T cells, you know, T cells get around. I think that was one of the big lessons that we had in terms of just seeing the image of infected cells moving as fast as they do, makes you understand how it permeates the immune system and is indelibly connected to memory responses. However, with coronavirus, you know, I think certainly when you have a severe infection, a high viremia, I think it's that's where you get more disseminated involvement of other organs. And it's not clear in that case whether it's the cell-free virus or some other, perhaps immune cells, which are ferrying the virus to different sites. You know, I think it could, the monocytes in COVID are very, are thought to be, you know, guilt by association with regards to the cytokines which are released. And, you know, they certainly could be spreading the virus to other organs, and there could be real significant compartmentalization. HIV is compartmentalized as well, but it's a different level of motility, I think, with HIV. But that's a really, you know, I find myself comparing the two infections all the time as well. Thanks for that question.
Ben, we could take one more question. Dr. Cespedes has a question around, if you can see it in the chat, the percent of infected cells secondary to cell versus virgin cells to cells.
And that's a really
not virgin cells. I meant viron to cell. I'm sorry. I'm just typing too quickly. Apologies there.
No problem. I'm actually.
And really, like, you know, it's a three-part question, but really I was thinking, Ben, you know, does that have implications for, you know, attachment inhibitors, the CCR5 inhibitors, maybe, if that viral synapse is so small, or, you know, does that make a difference? The attachment inhibitors look so efficient, but maybe later in infection or for these highly treatment experienced, might that make a difference?
Well, the attachment inhibitors are very interesting in that I think they could have unique effects on this cell-cell transmission, and also particularly like the small molecule attachment inhibitors like fostemsavir in particular. You know, we're interested because that particular small molecule has a ability to stabilize what's thought to be a more native state, native conformation of envelope. And I think anything that can stabilize envelope so that it's not fluctuating in its conformation as much as it as it does, I think has the potential to help the immune system to recognize it. So that's actually one of the hypotheses which we're pursuing with Dr. Aberg and studying antibody responses of patients who've been treated with attachment inhibitors. So I think there may be some role for things which interfere with synapse more directly, and you know, so those are or improved immune response. And I think it could be that certain classes of drugs can theoretically have, you know, I guess our hypothesis anyway is that it could limit the ability of envelope to access different conformational states.
Thank you, Dr. Chen. And so I want to thank you for grand rounds today for a very enlightening talk. So everyone, let's thank Dr. Chen and have a great day, everybody. Yeah, little claps out for Dr. Chen.
Thanks, everybody.
See you, Ben. Thanks a lot, Ben.